The last part we need to talk about with chapter 15 is what happens if DNA polymerase doesn't recognize it made a mistake, and then of the mistakes that persisted, the other machinery didn't recognize the mistake and fix them. And then so this is a mutation, right? It's typically a copying error that persists. Okay, it's a copying error that persists, and so that every future generation of that mutated cell now has a mutated copy of the genome. And so mutations come in a variety of forms. You have what are called missense mutations. And so missense mutations, they result in the wrong amino acid being put into the primary sequence of the protein. And so your missense mutation, a single nucleotide changes, and so it changes the codon, and that codon results in a different amino acid when it's translated. Okay? And so this would be a missense mutation. And here are some human genetic illnesses that are the result of a missense mutation. Sickle cell anemia, we'll see that in a little bit more detail in a minute. Albinism, which this isn't just a human genetic illness, you find it in almost all organisms. And so if you find a place where you've eliminated all of the selective pressure over animals, you get a lot of albino forms showing up. So you'll find like albino raccoons in the Midwest where people are like, you know what, I love the raccoons, they're so cute, I'm going to let them eat my garbage. And so you eliminate all the pressure, and there's nothing really eating raccoons anymore because people in Ohio killed everything that eats raccoons, except for people. Although people actually kill people in Ohio as well. Hemophilia, it's a genetic illness impacting one of the clotting factors, and so it results in an issue in the clotting cascade, and you don't clot properly. So it's a bleeding disease. And then achondroplasia, which is one type of dwarfism. Those are all the results of missense mutations. But I don't want you to think missense is always really bad. Because if you change the amino acid, but that amino acid is chemically similar to the one it was supposed to be, you're still probably going to have a normal functioning protein. Then you have nonsense mutations. And I love this. Like, it's a nonsense mutation. And you're like, so is that a real mutation or is it not, right? Is it fake? What are we doing here? And so this changes the codon into a stop codon. So it results in a polypeptide that's much shorter than it's supposed to be. I guess it doesn't have to be much shorter, because if it just moved the stop codon up one position, it's only one amino acid shorter. But usually results in a polypeptide much shorter than what it's intended to be. You end up with an incomplete polypeptide. Okay? This is a very high probability of impacting the phenotype. This, kind of an intermediate probability of impacting the phenotype. Okay, come on, welcome. And then another type, these first three are all called point mutations. And so the last form of point mutations are silent mutations. And they're not silent because it never impacts the phenotype. What it means by silent is it doesn't change the amino acid. So you have a mutation in the codon, but the codon still translates into the same amino acid. And this is because if you remember that chart of the genetic code, many of our amino acids have multiple codons that result in that amino acid, right? Right? Yes. Yeah.
Will a silent mutation affect anything at all?
Yes. Yes, they do. Yeah, so that's an excellent question. They're not going to result in a different amino acid, but they can interfere with the pre-mRNA processing, right? So they can interfere with where you read to splice out introns. And so you may look at it and you're like, wow, that doesn't result in a different amino acid. Why is this mutation so detrimental? And that's why I said silent doesn't mean it doesn't impact the phenotype. It means it wouldn't result in a different amino acid. But it could absolutely impact where your introns are, like where you cut that pre-mRNA as you're processing it. Yeah.
How does that even happen, though? Because they have to match. So if you have like an A with a G or something.
Oh, so how do they even make that mistake?
Yeah, they're not going to.
Because they're just the sheer speed at which they're replicating.
I mean,
Sometimes,
wouldn't there be like a bubble?
yes. Mm-hmm.
Okay.
Yep. Yeah, and so that's another way, Kinsey, that it impacts it, is just because it doesn't result in a different amino acid doesn't mean that the DNA molecule is good, right? It still had the wrong base added, so you're not forming hydrogen bonds in one place on that DNA molecule, and you have a bubble. And then it actually begets more mutations. So these are all point mutations. Yeah, Joey.
Okay, so just like you have like multiple codons, that could be one type of
Yes.
amino acid. Can
Yes.
you have multiple bases that can correspond to the same, like one codon, like you could change a base and it wouldn't change the codon? Is that possible?
Well, yes,
Seems like
because the
it's
third position in the codon is flexible, and so you have a feature that's called wobble, where you can change that third codon, and it would still attract the same anticodon.
And you wouldn't really have any type of mutation from that, would you?
Probably not, unless of course it changed the way your pre-mRNA was processed again. But yes, yeah. So these are all point mutations. The rest are, or I guess I'm just giving you one at this point, are called chromosomal mutations. And so if you have a frameshift mutation, like you delete a section of the template or you insert too much material as it's copied, it changes the entire reading frame and oftentimes will result in a non-functional polypeptide because you change the reading frame, so virtually every amino acid after that mutation is wrong. Virtually every amino acid after that is wrong. So again, these three are point mutations. This one is a chromosomal mutation. And there are a lot of types of chromosomal mutations. There are a lot of chromosomal mutations that'll change your reading frame and fall under this category of a frame shift. They're shifting the reading frame is what it's referring to. Does that make sense? Okay. So these are mutations and their impact on the phenotype is going to vary based on a number of factors, which we'll talk about in a minute when we take our lecture break. All right, so here's the normal gene. Here's the DNA. This is acting as the template strand, and then we go and write the complementary strand, but instead of writing a complementary DNA strand, we write a complementary RNA strand. So anywhere where you had a thymine is replaced with a uracil. But here's our mRNA, and then here's our start codon, and our start codon is always going to result in methionine, and then we just keep going on reading these three bases at a time and translating that into a particular amino acid. So here's our normal gene. Here's a missense. So remember, in a missense, we change the amino acid. Okay, we change the amino acid. So here, instead of having what was in our template there. So it was an A here in our template in our normal gene. Now it's a G, and so that G results in a different template, or it's a different template results in a different complement made. And so now our RNA complement is different than what it's supposed to be, and it's translated into a different amino acid. So this is a missense mutation. The codon changed and the amino acid changed. And I told you sickle cell anemia was one example of a human genetic illness that is the result of a missense mutation. And so here's the normal polypeptide and your beta globin, which is part of hemoglobin. Hemoglobin is a protein with quaternary structure. It has 4 different polypeptides combining. We talked about this, I don't know, when we talked about protein structure week 2. And so here's the normal amino acid, and here's your missense mutation resulting in a different amino acid. And so the protein functions differently and doesn't bind oxygen as well. And so, and it doesn't cluster as well, and you get weird shaped red blood cells that 1, don't carry oxygen as well, and 2, tend to clump together and form clots. Yeah.
So some of these mutations, like sickle cell and others, they have a big effect on the organism and they're quite noticeable.
Yes.
Is that caused by only one
Yes.
amino acid?
Right, and that's why you can't think of like the term silent, missense, nonsense. They're not referring to what happens at the phenotypic level, because that's going to be determined by a number of different factors. They're referring to what happens at the primary sequence level, right? And so in some of these, you may change the amino acid, but the protein's not going to have a huge impact on the phenotype. You just wouldn't even recognize what's going on. Mm-hmm. But then others, you're going to have a huge impact on the phenotype of the organism because of how important that protein is. Yeah, Kinsey.
Well, so like for instance, like mutation, it could possibly not have an effect on the phenotype.
Yes. Yep, absolutely. Yep, absolutely.
In what case?
Well, save that for a little bit because we're going to talk about that in our lecture break. And then some people around you may have some thoughts to fill in. Here's the nonsense mutation. And so again, a nonsense mutation changes a normal codon into a stop codon. So it ends your reading frame early. It's still a point mutation. It didn't shift the entire reading frame. It just stopped your reading frame early. Because once you get to a stop codon, you don't have a tRNA to bind there. Instead, you have a release factor that just takes your entire complex and breaks it apart. Yeah, listen.
Do any of these affect more than just one polypeptide chain?
Oh, yeah, certainly. Yeah, because remember that an mRNA can be spliced in different ways, resulting in different polypeptides. Right? And not only that, but polypeptides can be modified in different ways, right? Once you have a polypeptide, it can be cut into pieces. It can be modified in a number of different ways. Yeah. That's a good question. All right, and then finally, a silent mutation. So it's still a point mutation. Actually, it's not finally. I have to show you Frameshift as well. So silent mutation, you have a mutation there. It's the wrong base, but it resulted in a different codon that code for the same amino acid. So your amino acid didn't change. You still get the same amino acid. And this doesn't mean that it's never going to impact, which we'll talk about during our lecture break as well. Under what conditions will missense mutations not impact the phenotype? And under what conditions would silent mutations impact the phenotype? It's going to be fun. And then finally, frameshift mutations. And so in a frameshift mutation, you either insert an extra piece of chromosome or you delete a piece of the chromosome. And so it shifts the entire reading frame. It changes every codon, not just the one that bears that mutation. Every codon after it is changed. And so it shifts the entire reading frame. Every amino acid from the mutation on is potentially different. And you're going to oftentimes result in a completely different polypeptide. And many times you'd end up with a polypeptide that can't be folded. And then you could end up with some really, really bad possibilities. Yeah, Joey.
So like when the DNA is copying, is that just the polymerase moving so fast that it straight up skips one?
No. So with these frameshift mutations, they usually happen either if a virus inserts its genome into a gene, or a virus pulls itself out of the genome in the middle of a gene, or during crossing over. So like when two homologous, which we'll talk about a lot in a couple of weeks, when two homologous chromosomes line up side by side, they're similar enough that they'll actually form what you would call a tetrad, a structure with 4 strands instead of 2. And then there's a machinery that will fix that, and sometimes a piece of one chromosome ends up on the other, and they kind of trade some pieces of the chromosome. If that happens unevenly, some of them will end up with a shorter piece, some of them will end up with a longer piece. Either way, you're shifting your reading frame. On one of them, you have an addition. On the other one, you have a deletion. Yeah, additions and deletions are almost always going to be coupled with the other one. So you have an addition in one chromosome and a deletion in the homologous. So like your paternal chromosome will lose some, your maternal chromosome will gain some, or vice versa. Yeah.
Are these mostly caused by like external forces, or the other ones are like internal? Or then like they just read them wrong, or this is like an external force causes it?
No, because most of your frameshift mutations happen during crossing over, which is still an internal force.
Okay.
Now they can be also impacted by external forces, but so can point mutations, because point mutations don't have to be the polymerase made the mistake. You could actually modify it from radiation. Okay. Like radiation can actually change the base that's there. Okay. All right, it's lecture break time. So what I want you to do, again, we've passed exam 1, so you can assemble groups however you want, as long as there are 5 people in the group. 5 or 6, no more than 6, no less than 5. So you may still need to move yourself a little bit. And what I want you to do. Okay, hang on. You need to know what you're supposed to do. Although I did already tell you a little bit ago. So I want you to discuss. Shh. I want you to discuss under what conditions will a missense mutation not change the phenotype, and under what conditions would a silent mutation change the phenotype. Okay? Because remember, silent and missense, it's not referring to what's happening at the organismic level. It's referring to what's happening at the primary sequence of the protein level. Yeah, Alyssa.
Isn't a silent mutation a type of missense mutation?
No, because a missense mutation results in a different amino acid. A silent mutation results in the same amino acid.
Okay, so
Now, they could have the same impact, but that's what I want to know. Under what conditions would they actually be the same? Which there are conditions in which they would be the same. Yeah, Joey.
so missense doesn't produce mutation, and
No, it's still a mutation, but that missense doesn't produce something different at the phenotypic level. So like you right, so you're still
nonsense does.
right, you're making a different protein technically, but the protein's functioning the same. All right, go ahead and take a couple of minutes. I'll roam around and ask questions that hopefully will get you thinking.
So with the missense mutation,
Okay, so missense doesn't produce what? Missense not produce phenotypic change.
oh no, it's fine. It's okay.
And the nonsense does.
Okay.
I'll think small.
So, like get the food and then amino acids put in there. But
Yes, I mean we have tons of space.
Yeah, so usually it just changes.
oh no, the missense usually does. Because if you have a different amino acid, it changes the missense. But if the amino acid is close, then it doesn't affect the full structure of the protein, then it will affect the result of the protein. Okay, so normally missense results in
I mean, does it
so it doesn't have to, because if the amino acid
is it okay? I don't know. I mean, I'm not gonna, you know.
The amino acid structure.
I mean, you're not assessed really in any way in your performance in these discussions. So, other than like, you know, so here's where it may make an impact. And I know I'm taking away from your discussion. Let's say you just to a point at the end of the semester and you're like, you're at like something.
I've heard that it is, but just like have a big impact. So I guess it's not detrimental. Or even positive.
I said 5 or 6.
how does it not affect its phenotype?
You had 7.
And that way it's but and then a missense, not a missense, the silent one.
Nonsense.
You want to sit closer, Chris? Because you're trying to show me.
And then the
All right, you got about 20 seconds or so. Wrap up your thoughts. Get somebody to share your thoughts.
Go ahead. What do you say about the silent one?
So it sounds like you're affecting because if you do the total, it doesn't change. Like let's say you know there's multiple so even if the codon totally like affect a different amino acid, it can affect the structure of the actual DNA itself, which then can lead us to when it might later on be related, or it can affect ones that are later, or it can affect ones that are like it can like make them separate.
The DNA or the polypeptide?
The DNA can like separate. Hey,
I'm just talking with their niece over here.
pretty cool.
Yeah.
Which then can mess up like
Her and I are very similar, which is why we like to hang out and do nerdy things together.
Even though it's changes.
Alrighty. What do we think? We're gonna start over here. Is it because you guys have 7 and not 6, and so you could? Oh, okay. So you actually had 5, which is good, and 2, which is not good, and then made 1 that was not good. Okay. All right. So we're gonna start over here. Anybody have some ideas? Let's start with under what conditions would a missense mutation not change the phenotype? To where you'd have a missense mutation, but you couldn't tell in the way the protein was functioning. Alyssa.
a missense mutation could not affect the phenotype if the amino acid it codes for, even though it's different, is similar enough that it doesn't affect the structure of the protein and therefore it's functional.
Yeah, absolutely. We have these whole charts that show your 20 amino acids, and they group them based on how they behave chemically, right? You've got non-polar R groups. You have polar R groups. And then within the polar R groups, you have acidic and basic. And so these R groups behave differently chemically. So let's say you had a different amino acid added. You started with a non-polar. You kept a non-polar amino acid that was pretty much the same size, right? Because the R groups could be way bigger or way smaller in addition to being different chemically. You're probably going to get a protein that's going to function in the same way, right? The amino acid changed, but the overall chemistry of the protein is the same. It's going to be folded the same. It's going to interact the same. You're not going to see a real big impact at the organismic level, which is what we would think of when we think of the phenotype, what the organism is actually expressing. Good. Any other thoughts on how a missense mutation would not impact the organism as a whole?
Just good job, Alyssa.
Yeah, that was great. That was great. All right, so what about a silent mutation impacting what you see at the organismic level? Yeah. Yes. Absolutely. Yeah, so you may look at this mutation and you look at it and then you look at the normal strand and you're like, wait a minute, it's not actually going to the codon is different, but it codes for the exact same amino acid, and then you have no idea why the cell with that mutation isn't functioning, and then you remember, wait a minute, there's something that happens in between transcription and translation, and that's RNA modification. And so if it disrupts your splicing, you have a huge potential issue. I'll overlook the fact that you had 7 because that was a great answer. That was a great answer. I love it. Any other thoughts?
I have a question.
Yeah, Charles.
A silent mutation, could they affect anything else besides phenotype and therefore kind of deduce
Yes.
what introns and extrons are for?
Yes, sure, yeah. And then any other thoughts? I heard another thought from up here that just because it's not going to change the amino acid, it doesn't mean you added the right base. And so at least for a time, you added the wrong nucleotide and you're not forming those hydrogen bonds. So you've got a bubble until that's fixed, right? And you saw in here where we showed a silent mutation, something went in and fixed it. It was supposed to be an adenine, and the mechanism added a guanine, and something has to go in later and fix that, reading the template and now fixing the complementary strand. And so the mutation remained. So one, you have to expend energy to go back in and fix it, right? That's going to impact the phenotype. Energy expenditure going in and doing something that you shouldn't have had to do in the first place. And then another thing, until this is fixed, you actually have a bubble in your DNA molecule, which could fail a checkpoint. The cell could look at the DNA when it's assessing the quality and say, I've got a bubble here. There's an error, and actually trigger what's called an apoptotic pathway, where the cell kills itself because it knows there's something wrong with its DNA. Yeah, Joey.
Does it have any way of knowing, like, so it comes to, like, and it finds 2 that are wrong, to know which one was wrong, like which one was the original?
No, because it just matters which strand it reads.
So it's totally, so it can't, like,
It's probably not 50-50, but it's probably pretty close to 50-50 whether it corrects it to get what you had before the mutation or whether it preserves that mutation. Yeah, because it depends on which strand it reads. If it reads the new strand, then it's like, okay, we wrote the wrong new strand. That's its new template. It's going to change the old strand. But if it went back and read the old strand, then it's like, okay, we added the wrong base, and it'll fix it and put the right base in. Yeah. So, yes, it does matter which strand it reads, and it's probably pretty close to 50-50 whether it gets corrected or whether that mistake is preserved. And now that mistake is preserved, the cell doesn't recognize, at least unless it changes the protein, doesn't recognize anything wrong with the DNA molecule. All the hydrogen bonds are formed, there's no bubble, the DNA looks high quality. It's only if it's going to change the protein or change some kind of a regulatory element that the cell will recognize that there's an issue. All right, so here's another chromosomal mutation. How do transposable elements impact phenotypes? So you have these portions of the genome that move. They're flexible. They'll kind of bounce in and out of chromosomes, and we call these transposable elements. We We found them in basically every organism that we know a lot about their genetics. If we know a lot about the genetics of the organism, we found transposable elements in them. Now, if the transposable element moves from, say, chromosome 1 to a completely different chromosome, and not from your chromosome 1 from your dad into the chromosome 1 from your mom, that's usually not an issue, because they both have transposable elements and they'll just kind of exchange information. But if it moves from chromosome 1 to a completely different chromosome, you have another chromosomal mutation, another frameshift mutation, and this is called transposition. It's another example of a chromosomal or a frameshift mutation. Okay? So again, that frameshift, it's interchangeable with chromosomal, and it results in a shift in your reading frame. And so this can happen by two mechanisms. One is called cut and paste. And so cut and paste, you lost it from the first chromosome and you gained it in the second. Copy and paste is you end up with a copy in the old chromosome and a new copy in the new chromosome. And so you end up with two copies of whatever that transposable element is. And this is kind of exciting because now your old copy can go on doing whatever it did, whatever it was meant to do, and your new copy has the potential to maybe do something different. All right, so here's cut and paste. Again, the transposable element moves from one chromosome into a non-homologous chromosome. Say from chromosome 1 into chromosome 4, chromosome 5, a different chromosome. Not from like chromosome 1 from your mom into chromosome 1 from your dad. Okay. So we've got a chromosomal mutation. Cut and paste. Copy and paste, we've got still a chromosomal mutation, but we preserve the original. And so now you've got your original and you have your new copy, and you can get some really interesting things happening. All right? Any questions about this? I'm gonna skip the last few slides of this, which means you won't be assessed on the last few slides of this. And we'll transition over into chapter 6 on thermodynamics. Yeah.
What would you define a transposable element as?
A transposable element as what type of mutation?
Or just like what it is?
I mean, just a mobile genetic element. And sometimes you'll see them refer to that. So it's just a piece of the chromosome that's mobile. It's designed to actually leave chromosomes and re-enter. And then you're like, well, what's the function of that? We'll get there. But there's good function to it, but it has a potential to result in a chromosomal mutation. It doesn't always, but it has the potential to do that. Yeah.
Which one of the two is like more detrimental, the cut and paste or the copy and paste?
Neither. Yeah. All that means is like in some places you have the old copy and then in the other ones you don't. But what you're worried about is that new site. That's where you have the potential issue. And where you actually have the chromosomal mutation. Yeah. So they're both going to result in the new site, just one of them also has the old site.
Sorry,
Yeah.
Do any of these do this on purpose, or is it all just a mistake?
Transposable elements do it on purpose. There are actually machines that will cut these transposable elements out and then reinsert them somewhere else.
Okay.
Yeah.
It's not like a mutation.
Well, it can result in a mutation, but it's not there because of a mutation. Yes, Sydney. It's an excellent question, and we'll talk about that in a little bit. Not today. I'll give you I don't know, a little foreshadowing. Is it adds variation. It's a mechanism by which you can increase variation without having a larger genome. Yeah, we'll go with that for now. Any other questions before we transition to chapter 6? From 15 to 6, yeah.
Is there ever a time when an organism purposely mutates its DNA?
Purposely mutates its DNA. I mean, there are times where the organism seems to not care very much about whether or not it mutates because making more copies is more important than fidelity. But I don't know if there are times in which it does it intentionally.
Or intentionally changes its coding?
I mean, you could say that these transposable elements and that design is a mechanism by which the organism is intentionally changing its genome. Yeah. All right. important questions. So the first 4 questions we're going to deal with, I don't know if we'll get through all 4 today. We might. We've got a pretty good chance of it. What is energy? How does it transfer? What kinds of processes are spontaneous? What are the kinetics of ATP hydrolysis? And how can the cell fuel endergonic processes? And this one's especially interesting, and this one too, considering what we talked about with cell membranes, right? That active transport requires ATP. You have to cleave or hydrolyze ATP in order to facilitate active transport, pumping something against its concentration gradient. And so this is where we start to provide some of the chemical and physical explanations for how that happens. All right, so what is energy and how does it transfer? Since you're covering the same material in chemistry this week, you should be able to answer this question fairly easily. What is energy? The energy or the ability or the capacity to do work. And this is a fantastic way to think about energy because work means something very specific in physics. Work is you take a force and you execute that force across some distance, right? Something has to happen. Something has to move. And it's a wonderful way to think about energy. But it's not the activity of doing work. It's the capacity to do work, all right? So we've got potential energy, and it's like, yeah, I've got a certain capacity to do work, but it's not doing that yet. And then thermodynamics is the study of energy and how it transforms from one form to another or moves from one object to another without transforming type. This happens especially with heat, where heat will move from one object to another and you don't change the type of energy. All right, so this is where our laws of thermodynamics come in. And the first law of thermodynamics is in many ways probably the second most important when you're talking about biological organisms. And you're like, but it's the first, right? Shouldn't it be the most important? But the weird thing is actually not the first because there's a 0th law of thermodynamics, which is actually the first. So anyways, it's another story for another time. So first law of thermodynamics says that you cannot create or destroy energy, but only transform it from one form to another, or from one object to another, if you want to go that way as well. And I think, again, I think you'll see fairly easily why this is the second most important of the laws of thermodynamics with regard to living organisms. Shh. All right, so various forms of energy that matter to living organisms. Heat energy, chemical energy, electrical energy, mechanical energy, radiant energy. These are some of the energies that actually you find either transferring energy to living organisms or coming from living organisms.
And these all could do work then?
These all at least have the capacity to do work, yes. Mm-hmm. That doesn't mean they are actively doing work, because heat is really difficult to harness. Heat in many times is thought of as wasted energy because it's really difficult to harness heat to actually do work with it. Right? You're like, well, heat is nice because it keeps you from being cold, but it's a really difficult form of energy to harness and to actually use it to do work. Kinetic energy is energy of an object in motion. This is obviously meaningful for living organisms, at least those that are capable of moving. At least those that are capable of moving. And then potential energy is stored energy. The only form of potential energy we really care about with regards to living organisms is chemical potential energy. Is the energy stored in bonds. That's really the only kind of potential energy we care about with living organisms. You know, if you're anything like me, when you hear potential energy, you think of like a boulder sitting on top of a mountain with the potential to crush something underneath, right? We don't care about that kind of potential energy with regards to living organisms. You're like, well, Dr. Engel, what if there's something at the bottom of that hill that's going to be crushed? Then shouldn't we care about it? So energy can be transferred from the system to the surroundings and vice versa. And this is where, you get a little bit of a difference between what you do in chemistry and what you do in biology with regards to thermodynamics, is nothing in biology is a closed system. Nothing is a closed system. Like, we don't have closed systems in biology. Everything is an open system. Energy transfers, matter transfers. Right? You've got closed systems in chemistry. I mean, sort of. You can set up something that's pretty close to a closed system. But nothing in biology is a closed system. Everything is open. Energy transfers, matter transfers, and things are just messy, and it's wonderful, right? It's wonderful, because life shouldn't be predictable. And so the reason why that matters is oftentimes things happen in living organisms that you're like, wow, that shouldn't really work according to the second law of thermodynamics. But then you have to keep in mind that they're all open systems. So you have energy transferring in and out, and you have matter transferring in and out. And so really, weird things can happen. Like harnessing sunlight into chemical potential energy. Shouldn't happen based on the second law of thermodynamics, but it does, because materials are entering into the plants and leaving them, facilitating that happening. So here's a closed system. Closed system energy still can enter and leave the system, but matter cannot. Matter is trapped. And again, in chemistry, most of your reactions you set up as a closed system. You're like, energy can transfer, but matter can't, right? So whatever we start with, those are our reactants, and whatever we end with, those are our products. But the thing is, with living organisms, your reactants are changing literally every moment. Every fraction of a second, your reactants are changing. And every fraction of a second, your products are changing. And that's why nothing's neat. And nothing's super predictable. And then open system, these are biological entities. These are living organisms. We are an open system. Earth is an open system, right? Meteorites strike the Earth. Gases are absorbed by the Earth. Material leaves the Earth out into space, sometimes intentionally and sometimes unintentionally. The cost to put something into orbit is really decreased. Like 10 years ago, it was $10,000 a pound to put something into orbit. And now you can do it for just a couple $100 a pound to put something into orbit. Give it another 10 years and you could put yourself into orbit for just a small amount of money. All right, so then isolated systems, which isolated systems are completely theoretical. There's probably no such thing in reality of an isolated system, where neither energy nor matter can transfer. Maybe. Maybe.
What if you have like a really full, crazy room where nothing moves?
Well, so if you had something at absolute zero, so you had absolutely no energy.
Or you just have a room that's completely closed, and no matter
It's
how
not gonna
hard
work.
you hit the board, it's not gonna move.
It's not going to work.
Really?
It's not going to work. And I think even considering the universe to be an isolated system depends on how you view God's omnipresence. Yeah, which we could have some conversations there. All right, so here's energy flow that we care about of living organisms. We start with radiant energy that fuels everything. And radiant energy is pretty difficult to harness and fairly unorganized. And then producers take this energy and convert it into chemical potential energy, which is extremely highly organized and really easy to harness. Yeah.
Where does the sun get its energy from?
From a fusion reaction. Hydrogen, two different isotopes of hydrogen smashing together to form a helium atom.
Does it run out?
Yes. Yep. Yeah, when it runs out of material, when it runs out of deuterium is the main isotope of hydrogen, which has one neutron and one proton. That's the main one to fuel fusion reactions, is deuterium and another deuterium, or deuterium and a tritium, which is two neutrons and one proton. But yes, eventually it's going to run out of material.
How long do you think it's gonna take?
I don't know what the clock is. It's still in the billions of years that they think the sun can keep burning. Yeah, about 20 minutes actually. We will barely make it to the end of class. Yeah, before the sun will actually run out of energy, you'll have some kind of a thermal meltdown when you just get below an equilibrium point. But it's all theoretical, of course, because we don't exactly know. Nobody's ever watched a star melt down from close up. So, all right. So, what kinds of processes are spontaneous? And this is really what we care about with regards to living organisms. Like, it's nice to talk about, okay, well, we've got chemical potential energy, we've got kinetic energy, but let's actually see what kinds of processes that happen in living organisms are going to be spontaneous. And so, typically, processes that release energy or become more disordered are going to be spontaneous. And if it can do both, release energy and become more disordered, then it's a really spontaneous process. Did Prof. K talk about like pouring a puddle of water on a table and eventually it's going to evaporate? Even though that water will never get to boiling point, it's going to evaporate because in doing so it releases energy, sort of. Individual molecules do. The overall process actually gains energy, but it becomes far more disordered. And so it's a spontaneous process. And so these are especially meaningful with regard to living organisms. And so in reactions that happen in living forms, we care about how much enthalpy do the reactants have, how much enthalpy do the products have, and if the products have less, then you released energy in that process. And that is a spontaneous process, meaning one that will happen without the cell committing energy to make it happen. Bless you. Now, what's another thing we care about in addition to spontaneity? So spontaneity is good, right? If it's like, this is a spontaneous reaction, that's good because the organism doesn't have to invest energy to make it happen. Yeah, Kaylin.
Using energy to get something.
Okay.
As opposed to losing it.
Sure. Yeah. Speed, right? Time, right? There are a lot of spontaneous reactions that they'll happen on their own, but they're going to take a really, really long time. And living organisms do not typically have a really, really long time. Really, really long time. And so, exothermic reactions are, again, these that the products have less potential energy, less enthalpy than the reactants. So these are going to release energy. Endothermic reactions, the products have more potential energy, more enthalpy than the reactants, and so you had to actually invest energy into that. It absorbs energy. And so, more reactions that are, again, spontaneous if the products are less ordered, more random than the reactants. Have you talked about entropy yet in chemistry? And so entropy is technically the number of ways in which you can arrange the parts to get the exact same thing. Okay? So if you've got like all of the parts of a car just taken apart, there are a whole bunch of ways in which you can arrange those parts and still have a car that is taken apart. Right? But if you've got all of those parts assembled together, there aren't very many ways in which you can do that and have a functioning car. Okay, and so the car disassembled is more disordered than the car when it's assembled. And you're like, well, of course it is, right? You can see that. But there's your mathematical explanation for why. So this is where the second law of thermodynamics comes in. And the second law of thermodynamics, unfortunately, is oftentimes used as an argument against actually increasing complexity. And that's really unfortunate because that's not at all what the second law of thermodynamics is saying. So second law of thermodynamics just says that the total disorder of the universe is increasing. Second law of thermodynamics says the total disorder of the universe is increasing. It doesn't say that a system cannot become more ordered. It's just if a system becomes more ordered, its surroundings have to become more disordered to make up for that. Bless you. And usually what happens in the process is energy transfer. Is your energy becomes a little bit less organized in your surroundings. And so there are some interesting things that happen with the second law of thermodynamics. You could make the argument that the second law of thermodynamics makes the process of photosynthesis very unlikely. Okay? And absolutely you can make that argument, that the second law of thermodynamics makes photosynthesis very unlikely. Because in photosynthesis, you're taking a fairly random form of energy, sunlight, a fairly difficult form of energy to harness and do something with, to put to work, and converting it into something that's highly organized and very easy to harness and do work with. Okay? So the second law of thermodynamics makes that very unlikely, but it does not prevent it from happening. Okay? Because obviously it can't, because that happens. And so in the second law of thermodynamics governs the way the universe works. So you can't use the second law of thermodynamics to argue against increasing complexity. Yeah.
But in general, like, especially in the like, in terms of how life came about in the first place.
Yes.
Like, we see I mean, how do I put it? Life is one of the only things that can like reverse the entropy of a system like by itself.
No, I mean, living things are not the only systems that become more organized. They are probably the most familiar example and the things that do it most often. But there are all sorts of chemical reactions that the system becomes more organized.
Really?
Yes.
Can you give an example?
So a fusion reaction. A fusion reaction, you're taking two individual atoms and you are fusing them into a single atom. And so in that process, you are decreasing disorder. You're actually increasing order. And it is a spontaneous process for other reasons. It still happens spontaneously, but in terms of order, it actually decreases order. And then there's some things that have to happen in the surroundings. And so if you truly had isolated systems could not increase in order because there'd be no way to have net energy flow to compensate for that. But there are not any true isolated systems. So like closed systems could still gain in order. Open systems can do it easily because things, there are just ways to account for that. Yeah, so the second law of thermodynamics does not prevent things becoming more organized. Right? It can't, because things become more organized all the time. Right? That happens around us all the time. Things becoming more organized. And so the second law of thermodynamics obviously can't prevent that from happening. It does make it more complex, and it makes the origin of life issue a harder one to explain how it happened, because you're working against the second law of thermodynamics in some aspects, but it doesn't prevent it. Yeah, Charles.
2 questions.
Okay.
See, the first one, if we were to have, so we were taught that the universe is always increasing in entropy,
Yes.
and that means we're losing available, it has an implication.
Harnessable energy.
Harnessable energy,
Yeah.
energy is being lost, so like heat, like you're saying, that's a hard energy to harness. So if we had a truly isolated system where there is no back and forth, whatever,
Yeah.
and we were able to observe that environment, what would happen? Will it start to heat up and all our matter starts to disintegrate completely,
Yes.
and then it'd just be one big hot space?
Yes.
Interesting.
Yeah.
Okay.
And that's what'll happen if the universe is truly an isolated system.
Which also, okay, I got 3 things now. If the universe is infinite, how can it be increasing in entropy?
If the universe is infinite,
Yeah.
well, infinite in size does not really relate to entropy. So infinite in size is a difficult concept to just deal with in general. But as you start to fill more of an infinite space, you are decreasing in order. You're becoming more disordered. I mean, that can continue forever, right? But that's fine. I mean, it's just you're filling more of an infinite space.
Okay,
You
space
still
is-
have just as much space to fill as you did before, but you're still occupying more space.
Well, if there's infinite matter, if the matter itself is also infinite.
Yeah, you've got a lot of really interesting
But
things that you
that's-
have to deal with.
And then third of all, I've heard a lot of people, and they're usually people who aren't really educated in science, saying that entropy is a result of the fall, and I've always
It can't be.
disputed that.
Yeah, it can't be. And so Dr. Jones actually wrote a chapter for What Happened in the Garden, where he explained good entropy and bad entropy. And I think it's a meaningful distinction that you can see. Yeah, but I mean, and the reason I said the second law of thermodynamics is essential for life, we'll talk about that more on Wednesday. We're out of time. But yeah, it can't be. Yeah, entropy is not, it can't be a result of the fall. It has, I mean, it's just a way living organisms work.
Without it, the universe could not even exist.